clinically, what you can do instead is you can just go in and inhibit some of these myosins or pathways that regulate myosins, and those are really kind of super conserved pathways, and they're not really, not really subject to mutations the same way. And so what happens is you can reduce the forces. So here's those non-malignant cells, again, they make a little structure that look normal. Here's very little tiny nascent adhesions. There's those invasive kind of tumor guys with the large adhesions. If you inhibit forces, you don't make them become 100% normal, but you sure as to heck reduce their invasive behavior, and they start to look more like normal, so they've got little lumens here, and more importantly, you essentially normalize the behavior of these integrins. So I'm going to end here and just mention that we think tension is another way to imagine and understand that there may be modifications in tension that are important in driving, aberrant behaviors in tissues. We think tension forces play a role in as fundamental embryogenesis, normal tissue behavior. Every little aspect of your behavior of your body is regulated and governed by tension, from both extrinsic, so that can be the external matrix and things like crossing, but also intrinsic. And then in the case of cancers, either there can be an intrinsic behavior that modifies that can then change the cell, which can modify the matrix, or there could be something extrinsic that actually kicks in and modifies this and then kind of turns this around. The net result is that it modifies the behavior of the tissue and drives the kind of tumor-like behavior. And this is just a slide put together by one of my fellows, Matt, that really thinks about, disease. Is cancer really a disease of altered tensile homeostasis? Going everywhere from the extracellular matrix to the environment, and all the things that contribute to that. The immune cells, the fibroblasts, the vasculature, all these things can contribute to changing that. We didn't talk about the glycocalyx, but that's another aspect of it. The cytoskeleton, how the cells actually, respond, and various different oncogenes which change the cell's ability to respond to mechanical cues. And I didn't even talk about what happens at the different metastatic sites, but essentially what we think is that it affects everything from growth, survival, migration, tissue organization. We have some work on treatment response. We think it metastases, immune regulation. Everything is affected all across the spectrum, and so we have opportunities to explore there. And I'll just end with this slide, which really basically would like to throw out the idea that not considering force is like doing cancer half blind. So now I'm going to hand it over to Jan. Please come.
So this is probably the fourth time I've heard Valerie speak about this subject, and every time I learn a little bit more. And so, and it's hard to describe just how sort of different some of the things are that Valerie is describing in her own work. It really is what I think is a very interesting new view of some issues in cancer. And so I'm always, the more times I hear it, the more excited I am because it makes increasing sense to me. So what I wanted to do, rather than go into exquisite detail about any one aspect of biology and physics, I just wanted to daydream very generally about this intersection of physics and biology and physics and cancer in general. Because initially when you tell someone, oh, we're bringing together physicists and cancer biologists and we're somehow going to address problems in cancer, the gut response, of course, is, does that make sense at all? Isn't that a crazy or silly idea? And I agree if that were done in isolation. If you could bring 10 physicists together and ask them to address a disease, that I think would be a very silly undertaking. And so what's really special, I think, of what this initiative is trying to do, it forces people like Valerie and myself it forces us to interact daily. And that really is, I think, what will ultimately make this all come out right. So just zooming back about 100 years, I wanted to think briefly about sort of what kind of problems physicists and engineers have really been fascinated by in biology. And so if you actually go back about 100 years, biology involved chemistry, geometry, and physics. And a classic textbook is Darcy Wentworth Thompson's book, 1917. And he was interested in understanding all the beautiful structures he sees in biology. So he was a typical English naturalist who wanders through New Zealand, various other countries, wanders along the beach, studies various shells and honeybees, and he studies the wings of flies and so forth. And the thing he didn't understand is why a lot of biological materials have very regular patterns. And so, of course, if you look at a sheet of epithelial cells under the microscope, you'll also see that the cell shapes are rather regular. And when he looked at this initially, the first thought he had, oh, this looks like a collection of soap bubbles. And he spent a long time trying to figure out why there was this similarity in form. Other things that physicists love to ponder are, for example, here's an example of a vegetable that we can all buy here in the Bay Area. This is a particular kind of broccoli. And if you show this to physicists, the first thing they'll see is, oh, this is a particular mathematical series that actually determines the detailed shapes of the little florets of this broccoli. And why should it be that very basic mathematical expressions are displayed in this particular plant? Likewise, of course, we can. Some of my colleagues spend their lives wondering about snowflakes. So why is it, for example, that if I take a substance like water, if I cool it, in certain situations I can make these beautiful snowflakes, and notice that they're symmetrical. And when we think about the snowflake forming, of course, water molecules are being attached to the ends of the structure. But how on earth could it be that water molecules being attached to this side of the snow crystal, and the crystal is growing, do so in a way that is symmetrical with the water molecules being added over there, and over there, and over there, and over there? So how can it be that this regular structure grows over time? And so these are all traditional questions at the interface of physics and biology, this relationship between typically form and function. How are those related? Now zoom in, fast forwarding 100 years. So this is now mature biology, right? So what have we had over the years? We've had this enormous explosion of molecular biology and biochemistry. We have physics largely disappearing. And this field of biophysics emerges. It has a traditional focus on instrumentation methods, X-ray crystallography, NMR, and so forth. This entire era, the last 50, 60 years, I think, have been really dominated by the gene. And there's this idea formed in the 1950s that all the things we know about. And the key idea, I think, that's been driving us as a field generally for about 50 years is this notion that if we could only sequence the entire genome, then we could read this entire blueprint of life. So once we know what all the genes are, and once we know how many they are, once we know which ones are mutated in which cancers, then all the mysteries will have been revealed. And the issue is, of course, the human genome was sequenced in '99. It's now more than 10 years after that. And the question arises, have we solved every problem in biology? And the answer is clearly no. And so despite us now being able to completely read this blueprint, it's not actually clear to us how to translate that information into things that we care about, like diagnostics and treatment and so forth. And so here are some of the issues. This is a little snapshot of a protein interaction network that's important in embryonic stem cells. And I'm sure you've seen many networks like this before. You have colored dots, and each colored dot is a protein. Then you have arrows connecting these proteins. And if I now plot a similar network, this is a small snapshot of what we now know for cancer. This is a tiny subset of what we know about signaling in cancer. And if I were to create a comprehensive map like this, it would have easily 1,000 proteins on it. And it would have on order of 10,000 arrows. And so now just imagine for a moment in the situation of having about 1,000 proteins on there. The most important ones, there's maybe 20, 30. And all these arrows. And then imagine taking a small molecule and hitting one of these things. And so then the question arises, is that